Thank you so much, Clara, for nice introductory words. I also want to say thank you to the organizers for inviting me to this prestigious series of lectures running for many years now and having had enormous impact on the psychiatric culture here in the Stockholm region. I also say thank you to all of you who, challenged by wind and snow, have made your way here. This slide is for transparency, so here we have my affiliations. And I have to say that I had to revise the lecture for today, and I will include a few slides by the beginning, simply because of what's going on in our region, and comment a little bit on this news that came out almost 2 weeks ago. As we know, AstraZeneca will close neuroscience in Södertälje and leave off about 1,200 people, and this has of course been given high attention. It has impact on the region, and since you sitting here represent a considerable part of people active in this area in the region, I think it's very useful for the future to share a few thoughts and views. This slide is for industry in total. It illustrates the enormous increase in spendings in drug discovery and development over the last few decades, as you can see by the orange part here. This was a successful business up until the turn of the millennium, with a parallel increase in number of new drugs on the market. But as you can see, something dramatic has happened over the last 10 years. Despite continuous increase in spendings, there is a considerable drop in the number of new drugs on the market. This can be analyzed, viewed, and discussed from many angles, I think for hours. I will view this from only one angle, and it will not be the full picture. But I think already from this slide you can see that this is not sustainable. We cannot have such investments. The common number given for investment of a new drug in general is 1.3 billion US. By itself, a high number. This is probably an underestimation. If we instead divide the R&D budget of different companies with the number of new drugs produced, we end up with a cost per drug between 4 and 12 billion USD, depending on the company. So it varies considerably between companies. What this has to be balanced with is the annual earnings for a new drug having a patent life of 10 years. And we can understand that this industry is under considerable threat. CNS disorders has since long been viewed as a challenge in this regard. As you can see here, the success rate for CNS is below average. This is a reason for why, in particular in the CNS area, there has been a strong drive to find biomarkers of disease or biomarkers of mechanism to demonstrate that the drug binds to the intended target in the human brain. One of the examples of such investments which were initiated in academia is the development of new radioligands for amyloid, fibrillar amyloid detection in Alzheimer's disease. This started some 10 years ago and has had a major impact on research in Alzheimer's disease. I can mention that the radioligand here, PIB, very recently has been used to confirm 2 antibodies from different companies, but both confirmed to reduce amyloid plaque burden in the Alzheimer brain. So that's an example of a good application of a biomarker. In the collaboration here between AstraZeneca and the PET Center at the Karolinska, we aimed for development of an improved radioligand, more sensitive for detection of amyloid in the human brain, and have actually developed two such ligands, one labeled with a short-lived radionuclide, 11C, primarily for research purposes, one labeled with a more long-lived radionuclide, fluorine 18, having commercial potential as a radiodiagnostic. And this development has been successful. Here you can see detailed visualization of amyloid plaques in the human brain. And I think I can say that this is by far the best radioligand developed so far for this purpose. And have a PhD student working on this, among other things, interested in trying to relate local amyloid deposits to cognitive deficits. So this is very much ongoing work. But, and here comes the critical point. And I would say, if I was a neurologist, life would be very easy, wouldn't it? It's possible to visualize the disorder. Nothing new. Amyloid was identified in the human brain back in 1906 by Alois Alzheimer, so that's not new, but it can be visualized and seen. It's more tricky to be a psychiatrist. We don't have this type of landmarks. We have to search at a different level. But despite this progress in the Alzheimer area, we see that industry drops out from Alzheimer's disease as well. Not only psychiatry, also neurology, because again of the costs associated with finding new effective treatments. But psychopharmacology is worse off. That's the fact right now. AstraZeneca moved out from psychiatry 2 years ago, the preclinical studies. Novartis a month ago, GSK 2 years ago. Merck has reduced activities, so has Sanofi. The list is long. I discussed this with a guy who has perspective on European drug industry earlier this week, and he estimated that investments in psychiatry from pharma today is about 30% of what it was in top years. Of course, this has impact. A part of the problem may be the complexity and the challenge we are facing. As you know, major classes of drugs, antipsychotics, antidepressants, were discovered in the 50s and 60s empirically. People were sensitive to unexpected observations and turned them into advantages. Now, at time of rational drug design, identifications of multitude of proteins in brain, the question is where to go. Which protein is related to pathophysiology? And industry cannot afford anymore to make long shots in darkness. There has to be some understanding of pathophysiology, or at the endophenotype level at least, to provide some directions. So I think what we face here and aim for is a scenario for the future where research on the pathophysiology, clinical research, primarily will be a societary task. This is not to say that the way I read it, that industry is not interested. If by good research on pathophysiology it's possible to identify new targets, investments will be made, because as we know, there is enormous suffering and unmet medical needs in this area. Coming back then to what will happen now. AstraZeneca will not move out completely from neurology and psychiatry, but this will be run in a different fashion by something called virtual neurology, which will be based in Boston, 25 people. Virtual means simply that they will not have direct wet labs in-house. They will work through CROs, partners, elsewhere. There will be also a small group in Cambridge, 15 people, and there will be a translational science center here at the Karolinska, with a core of the PET facilities and also some people working closely with Science for Life, a few more. That will be our core. And I think it's good if we see this as an opportunity. Can we build translational science in this area together in a collaborative fashion? Then I think we will be identified in this dynamic network between Boston, Cambridge, and the Karolinska, which can take us, if not anywhere, so it's a good starting point to build again. Okay, with that I move to the topic of today. If it's possible to image the soul, and as you understand, I use imaging here as molecular imaging, or PET. First, the definition of the soul. The classical definition, which has not been debated very much, it has been taken for what it is for centuries, is that the soul is about intellectual life, tankeliv på svenska, volition, vilja, emotional life, känsloliv. This is not very much disputed. And you can find this definition 1,000 years ago. What of course is more disputed is the nature of the soul, but I leave that for the future. How did this start, or how did it start right here? To approach this question, how to relate brain biochemistry to higher brain functions. It started by an unexpected observation, I must say, back in the late 1980s or mid-1980s. We made a big study in control subjects who were examined, each of them 5 times, to determine Bmax, the receptor density, and KD, which is affinity. This was for them to be a good comparison group to neuroleptic naive patients with schizophrenia, such as the ones Clara referred to in the introduction. Something unexpected happened. After the higher doses, 300, 400, 500 micrograms intravenously, some of the subjects, yes, rather many, became restless. They showed motor restlessness during the PET study, started to move their legs, and also reported subjectively a sense of unease, restlessness, or distress. For those of you who are clinicians, you know that this is akathisia. It was a classical syndrome of akathisia, and we did video recording to document this, because it was found in the vast majority of subjects. Interestingly, it looked like this. So this is scanned from an old glass-mounted slide from those days. They have on the top, if I find a way here, the total uptake in the striatum, the putamen. We have the uptake in the cerebellum, and the difference here is specific binding, depicted by this curve. And here you can see motor restlessness coming close to the peak of binding or occupancy, and then disappearing. Similar for reported restlessness. The pattern looked different in different subjects, but it was kind of fascinating here to observe a subject who had an emotional manifestation, which restlessness is, and at the same time to follow receptor binding in brain. And another reason for giving this example is that it has clinical applications. This may appear as a sidetrack, but it is so that akathisia is one of the most overlooked side effects during antipsychotic drug treatment, often misinterpreted as agitation, rendering higher doses, new injections of antipsychotics. Akathisia appears within minutes or at oral treatment, 30-60 minutes after injection. So if you're an emergency doctor, then keep an eye on restlessness. The next unexpected observation came from the fact that the number of receptors in brain varied considerably among this comparison group of 10 males and 10 females. And together with Erik Jönsson and Peter Gustafsson, we related density to scores of the Karolinska scales of personality. And many of you have seen this data presented before. What we see here is a negative correlation between detachment, one item of the 15 of the Karolinska scales of personality, and D2 density. So if there is low density, then high score on detachment, which is viewed as a dimension with one extreme being people who are avoidant and cold and withdrawn, the other dimension people who are clingy, adherent, never leaves others alone, and a part of the normal flora of personality and behavior. So this was given attention and was a basis for a research strategy, one of the bases for the research strategy of the Stockholm Brain Institute when the discussion started about 10 years ago. Namely, that we can view molecular imaging here as providing a link in the complex chain relating the genetic endowment to human behavior, including higher brain functions. So this became a part of the strategy of the Stockholm Brain Institute. Of course, we can ask ourselves, why so different receptor density among human subjects, healthy controls, normal human subjects? We don't fully know. And we can speculate on genetics, age, environment, nothing new here, all discussions, and potential disease effects. We know that there are genetic contributions. They do not fully explain the variability, so this is a part of the story. Moving on with new generation of PET systems, we were able to examine a larger range of regions expressing dopamine receptors. And this was brought together by Simon Chervenka in his thesis in this figure, which we view as the spaghetti plot. What we have here is the mean binding potential in each of 8 brain regions, from striatum, thalamus, insula, and so on. And then we have plotted each individual subject, how much they deviate from the mean. And as you can see here, if you are high in one region, it does not mean that you are high globally throughout the brain. It's more like an individual fingerprint. And the challenging question here, of course, is this fingerprint caused by genetics or environment? Is it a mirror of the life history of the subject, or to which degree is that possible? And how does it relate to function the way we are? And you can see the emerging complexity here. We have an ongoing twin study. It has been concluded, and we are in the phase of analyzing the data. So I'm sure that in the near future, new light will be shed on this basic question. But I jumped the twin study for the time being until we have written up the manuscript. A way to approach this particular problem to some degree was in an analysis made by Simon again in his thesis, based on observations in vitro, in experimental animals, that the striatum is organized to a much higher degree than thought of. So applying this approach to PET data, Simon could demonstrate that there was significant correlations between the level of D2 receptor binding in the ventral striatum and tests measuring episodic memory, whereas no such correlations were observed for the somatosensory part of the striatum. Again, illustrating that there is a richness of information to arrive at and analyze in this area. So far, everything has been about biochemistry being the determinant and driving behavior. What about the other way around? And I mentioned the environment already as a possible player here. But we were not able to approach that fundamental question. Can environment, external stimuli, have an effect of brain chemistry? Until we started some discussions within the Stockholm Brain Institute. This slide is a couple of years old, but basically the same composition nowadays. So in initial discussions, we started to think of working memory as a way to address the possible influence of environment on brain biochemistry. Working memory, because it has since long been known to be related to dopaminergic neurotransmission. Listed some of the classical studies here. Goldman, Rakich, Patricia, not to say the least, has contributed enormously to this field. Patrick Klingberg in the Stockholm Brain Institute had at that time successfully developed a training program for ADHD, training of working memory, documented effect and relation to brain activity as measured by functional MRI. So the question arises in this setting, can we understand this further, the biochemical underpinning, by combining what already has been done with PET imaging of the dopamine system? We embarked on a rather elaborate experimental program at baseline, performance testing, functional MRI, 2 PET ligands, one for the D1 and one for the D2 system. And then the subjects, 13 subjects, underwent systematized working memory training for 5 weeks, and the entire procedure was repeated. This is a rather busy slide, but to the left here, we can see that as expected, there was an improvement in working memory. The cortical regions previously found to be related to this improvement were improved also in this study, so consistency here. For the D2 D2 dopamine receptor, there was no change, no correlations. For the D1 dopamine receptor, there was a relationship. And to describe this simply, those subjects who had high D1 density at baseline, they moved to the mean. Those who were low at baseline, they did also move to the mean, but in the other direction. Is this strange? No, it was not very unexpected. This is simply the reference. Because an inverted U shape has been described in the literature, both in the experimental literature and also at the same time in imaging studies. So here we have a relationship, and again, this is cross-sectional. This is not a challenge study, no training, but simply cross-sectional. Performance in Viscon's card sorting shows a U-shaped relationship with D1 receptor binding. So there's some consistency here in the literature. So to conclude, working memory training has an effect on brain biochemistry if D1 receptor is used as a marker, but not so on the D2 receptor. Are there other observations pointing to, let's say, flexibility, adaptivity of the part of the dopamine system defined by the D1 dopamine receptors? So this made us think a bit more. There are some indirect such observations. One stems from test-retest analysis of two of the radioligands we frequently have used for D1 receptor imaging. This is NNC112, and this is the older ligand sharing 23390. And typically, when we do test-retest with these ligands on the same day, there is a bit over 10% difference in binding potential. This is not seen for other well-behaved radioligands such as raclopride or WAY10635 for the serotonin system. So why this higher test-retest difference for the D1 receptor? We could not find an explanation from the characteristics or behavior of the radioligand. So could it be that this has to do with biology? A change over the day in density expressed in brain. Another observation, which is not fully in line with this, but again points to, let's say, flexibility of the D1 receptor system, is on the age effect. For most ligands, markers for the dopamine system, we know that we lose between 8–10% of the markers per decade. And I used to take this as an excuse for not being able to beat my sons in Nintendo, since I've already lost a good number of dopamine neurotransmission. But that's a sidetrack. That's rather typical. The question is, what happens before adult life, before 20 years? And one of them who addressed this question was Phil Seaman, who back in the 1987 did a postmortem analysis and suggested that there was very high density of D1, D2 receptors at age, let's say, 7, 8, 9, and then a dramatic drop during adolescence, followed by a much, much more slow decline. And this is then in line with the theory of synaptic proning, we have an abundance of synapses in 6 to 8 years, and then we lose the ones we don't use. So that was fundamental theory and still is. We had an opportunity to embark this question empirically in the human brain by having 2 control samples from adolescents, one from a study in schizophrenia and one in ADHD. And as you know, trajectories during adolescence are given high interest in child psychiatry and adolescent psychiatry, not to say the least from Judith Rappaport's group. So we examined those, and indeed, we found that during adolescence there is a much more rapid decline in D1 receptor binding than during adult life. So this is then in line with Phil Simmons' observations post-mortem, and also speaks to the D1 receptor defined system as being subject to changes over time, but in this case with age. More has been done with the D1 receptor, and I will come back for a minute to pharmacology. The radioligand here 23390 is not very useful as a test drug for treatment. But preclinical work suggested that D1 antagonists, they might be useful for the treatment of schizophrenia. And a company Shearing Plough in New Jersey developed an analog to 23390, 39166, and initiated clinical development. And we made a rather large clinical study here in Stockholm together with Uppsala, included, I think it was 17 or 18 drug-free patients with acute psychosis. And this was in an open study, but it was in a setting where we previously had examined other antipsychotics and at least could see if they were promising or not. But this was a big disappointment, because if anything, the patients went worse. We had to withdraw a good number of patients because they became worse and were not possible to maintain in the study. No observation of improvement. And this was very consistent with similar observations in Holland. A group in Groningen made a similar study. So first, this tells us that the D1 antagonist story was not translatable using the animal models predicting antipsychotic effect, or I should caution it by saying, since Torgny is here and has a lot of views on this, that the way they used the animal models may not have been correct. But it also may, if we put this in perspective, we may ask the question, is it the right thinking to view proteins in the brain as something you either stimulate or turn down? Or could it be that what we need is rather drugs helping us to find a balance, the way the subjects did who underwent cognitive training or working memory, finding a level at which performance was optimized? I think we have reason, and coming back to industry, has a reason to think of how the brain works in a different fashion than in the past. So, what do we do then for the future? 2 years ago, we said, one thing we face all the time when we try to make this type of analysis is the problem of our small samples. 10 individuals, 12, 15, separate studies. And that type of sample size is often too low to go into details. So what we set up, and this is informal, this is not a formal name, but we refer to it as the KI Behavioral PET Database. So we have started in a systematic fashion to bring in subjects again, examined in the past by PET, and in a systematic fashion draw blood for DNA, administer a standardized cognitive test battery, and do personality assessment. And to our surprise, or very much thanks to students working hard on this in the lab, we now have about 100 subjects examined with raclopride, equal number on sharing, over 100 on way 10635, wrong spelling here, a serotonin 1A receptor, about 50 on the serotonin transporter, and we continue to find more subjects here. This sample size is more or less unheard of in the history of molecular imaging. So this is an interesting database to start to look into, which is what we are doing, and having some very promising and interesting initial findings. And I will share with you today simply one new and yet unpublished observation, which is on diurnal variability. In the past, we have asked if there is a diurnal variability of D1 receptor binding, but it cannot be examined with 6 subjects. Here we see a clearly significant variability over the day when we examine 90 subjects. So from this it seems as if we have higher D1 receptor density in the morning than in the afternoon. Is this right? Can this be understood in any way? Well, it's tempting to go into the theory of receptor trafficking, which is an area developed very fast over the last decade, saying that receptors are mounted in the membrane, fully functional. There are mechanisms by which they can be internalized into the cytosol, some degraded, some recycled and used again. Could it be that such mechanisms operates at physiological conditions over the day? The D1 receptor is not very sensitive to endogenous dopamine. So there must be other mechanisms. So could this be the first direct evidence of receptor trafficking operating in the human brain at physiological conditions? That's the way we want to position it. But if so, that may make us rethink how we view the brain. 20, 30 years when I started, my conceptualization was that of a rather static organ, static levels of receptors or other proteins. Could it be that adaptive mechanisms from stimuli and what goes on during the day do not only influence the level of neurotransmitter concentration, but adaptation may also be a consequence of rapid, efficient receptor trafficking? Or could it be that such mechanisms, the adaptiveness to the external world, is something that can be handled different and may distinguish control subjects from subjects with major psychiatric disorders? These are speculations, but to take advantage of the enormous progress in basic neuroscience, we have to find ways to implement that in direct imaging of the human brain to bring the field forward. Answer to the question posed. I think it's possible to measure the effect of soulish activity on brain neurotransmission systems in many ways. If we by that measure the soul or not, I refer to as a philosophical question. Many people have contributed to this work over many years. I list a few here. More names could be added. But I do it this way also to emphasize the strength and power of groups collaborating across. Because important contributions here originate from all the groups listed here, and there are many more. So with that, I think I've used my time, and I thank you for attention.